Hello, everyone. We're delighted to welcome you to the quarterly HCA Town Hall. To make it easier for everybody across time zones, we're holding two identical sessions this time around, and you're in the first one. This session will be led by me together with the vice chairs of the organizing committee, Chloe Villani and Haniffa. Here's what we'll cover today: recent and upcoming HCA activities, goals and requirements for completing HCA version 1.0 atlases. And then Chloe Villani will present the recently approved HCA Immune Atlas, which is part of that HCA version 1 for all the different bionetworks, tissues and organs and systems. And then finally, we'll have our vision for HCA 2.0 steps, followed by a Q&A and closing remarks. The town hall is designed for open discussion, so we encourage your participation. Please submit your questions and comments in the Q&A section anytime. We'll try to address them as we go, and we've also reserved time at the end for further discussion. While most of us track time by the calendar year or perhaps the solar or lunar year, a human cell atlas year is defined by our general meetings. And we're grateful to everyone who joined us at the 2025 general meeting in Singapore this past June, which was amazing. You can see the energy on the slide. Was a lot of people, fantastic talks, amazing exhibitors. And we're already looking forward to welcoming you to the 2026 general meeting in Boston next June, 16th of 18th of June, 2026. Looking at the year ahead, so from the last general meeting, we reached the midpoint of assembling version 1 of the HCA Atlas, with draft atlases completed for 9 biological networks. And our next milestone is to have all 18 version 1 atlases completed by the time we meet in Boston. At the same time, we're preparing for the next phase of HCA. As we approach our 10th anniversary, our ambitions are becoming both broader and more global. Back when HCA began, it was just becoming possible to profile tens of thousands of cells. But we set our sights, even back then, on tens, hundreds, or a billion of cells. And these numbers are now becoming a reality, especially thanks to spatial technologies. So we can really accelerate Atlas expansion with more integrated data across modalities, across tissues, and across organ systems in 2D and 3D. Equally important is expanding the diversity of the Atlas so that it really reflects the breadth of human biology. Ultimately, our goal is a high-quality, healthy reference atlas that advances how we understand and treat disease. Here today, we'll share a survey with which many of you contributed to, and your feedback has already been invaluable. What comes next? Between now and June next year, there's important work ahead. Think of this as a collective call to action, and there are ways in which everyone can contribute. You may be contacted by biological network coordinators to provide data or metadata, because our data ecosystem team has streamlined the ingestion process and will guide you through it. So please do respond to these requests, because they are a crucial part of building the Atlas. If you'd like to participate in community-driven cell annotation, there'll be plenty of opportunities for you to join annotation jamborees on the cell annotation platform organized by the biological networks. So looking ahead, you can also get involved in HCA version 2.0 efforts, particularly through the Spatial Omics Task Force. To make sure you don't miss the opportunities, please keep your Human Cell Atlas membership profile updated with your current email and research interests. Later in the town hall, we'll share more on how to stay connected with the HCA community. Upcoming meetings. So I've mentioned the next HCA general meeting will take place in Boston at Harvard Medical School, June 16th to 18th. It'll be hybrid and registration is already open. We hope to see many of you there, whether in person or virtually. Beyond that, we're excited to announce our future HCA Asia meetings, and in 2026, we'll meet in Perth, Australia, and in 2027 in Jeju, South Korea. Please stay tuned for the exact dates and the registration details in the coming months. As always, we are grateful to both our past and future sponsors, and 10x Genomics is a foundational sponsor for the 2026 general meeting. If anyone is interested in sponsorship opportunities, please do contact us at meetings@humancellatlas.org. Many of you have heard the exciting news at the March Town Hall and in Singapore about our new partnership with UNESCO, which is the United Nations Education, Science, the S is for Science, and Culture Organization. Together, we've identified strategic priority areas that align closely with the HCA's mission, and we are so proud to forces with UNESCO. So the priority areas are open science and equitable data, policy dialogue and ethical frameworks, capacity rebuilding and regional inclusion, and cross-initiative integration and synergies. We'll keep you updated through our website, events and news pages, newsletters and social media on UNESCO activities. Include an exploratory webinar series on single-cell genomics and the exposome, and we already, of course, had some nice topics on that at the Milan general meeting, where we had the organoid and environment working group, a joint bioethics working group between Human Cell Atlas and UNESCO, single-cell capacity building workshops, and supporting open science, science diplomacy, and public engagement. So very exciting. So now let's move on to the next topic on our agenda, which is the completion of an HCA version 1.0 draft atlas. We'll take a closer look at our goals and requirements for this important milestone. First, we want to congratulate the teams whose V1 atlases have already received official approval from the HCA Organizing Committee. More recently approved are kidney, gut, development, immune, and liver cell atlases, and they'll soon be available through the HCA data portal. And just for everybody to be clear, it's data.humancellatlas.org. So very simple link for the Human Cell Atlas data portal where these data objects are available. The kidney atlas integrated and annotated 18 single nucleus and single cell data sets, and the resulting integrated atlas contains 1.7 million single cells and nuclei representing both healthy states and disease conditions such as acute and chronic kidney injury. The gut cell atlas comprises 1.5 million cells from 27 data sets and 288 different donors, including data from Helmsley Charitable Trust Crohn's Gut Atlas groups. This remarkable effort brought together 12 labs for annotation, a true testament to the strength of HCA's collaborative spirit that guarantees super high quality and expert data releases. The Human Developmental Atlas includes over 3.6 million cells from 188 donors. The team integrated developmental data from 21 organs between 4 and 22 post-conception weeks, so first and second trimester, incorporating published data and newly generated whole embryo data. And in a minute, you'll hear about the Immune Atlas. The Immune Biological Network collaborated closely with the Genetic Diversity Network to bring that work to completion. And that's PBMC, cells, peripheral blood mononuclear cells, which is really crucial also in terms of understanding what is healthy reference blood, and as a reference, because blood is key diagnostic tissue, and this will no doubt become an underpinning reference for right across all the different biomedical communities, I'm sure. So very excited that Chloe will be presenting this later today. Over the next few months, the oral and craniofacial and the pancreas biological networks will undergo evaluation for approval by the HCA Organizing Committee, and will continue to update you as additional HCA atlases are accepted and made publicly available on the data portal. So once again, the goal is to get the V1 reference for all the 18 bionetworks by the general meeting in June 2026. And once again, we want to extend our sincere gratitude to all the teams around the world who are working collaboratively on these efforts. Each completed atlas represents years of community-driven work and shared commitment across data generation, data analysis, data cleaning and serving, and it's just so gratifying that we've arrived here today. This slide is a reminder of the established guidelines for an atlas to be accepted as an official version 1. So just to remind you, robust technical factors includes all landmark studies, is driven by the biological networks that are coordinated by our leaders in each field, and all the PIs and group members that contribute to that. The annotation is therefore with expert input from the biological network members with the analysis, data analysis colleagues. The annotations are in the cell annotation platform, and we make of not only the integrated data, but also metadata and the code publicly available on the data portal at data.humancellatlas.org. And we continue to announce the releases via our social media channels. So please do keep an eye out for those updates and feel free to re-amplify the message out to the community. So what sets the Human Cell Atlas apart from other initiatives building tissue or organ specific atlases? To emphasize the importance of the collaborative work to achieve HCA version 1.2 completion, each depends on community input, and each is driven by the biological networks with colleagues. So one is HCA atlases that have a huge scale. So 100 million cells, both in terms of the scale of the data and the scale of participation of scientists who are experts in each of the biomedical areas, and of course in data analytics, data science, AI/ML, from over 100 countries. And the Human Cell Atlas community, of course, draws on that diversity that we're absolutely committed to, both in terms of our data and our scientists, to building representative cell atlases to the best extent we can. We're also committed to a broad scope, full body coverage, aiming to Human Cell Atlas, enabling cross-tissue analysis. We're also committed to flexibility and to the biological nuances, but staying aligned to technical standards and data standards across the different tissue atlases. And so that really emphasizes our methodological rigor, that we are continue to remain at the forefront in terms of both the data generation technologies and the AI/ML and analysis technologies. And methods are continuously being advanced and tested by the bionetworks and the computational community. Finally, I want to really emphasize our participatory spirit. Anyone can be involved. We welcome people across the globe, across scientific disciplines, and all our teams are cross-cultural in a scientific and in a country sense. And so the aim at the end of the day is to build the highest quality, most representative and most. So the goals for the completion, once again, are to publish the source data sets for integration. So all Atlas source data sets, the tier 1 metadata, are on the HCA data repo and CAP, necessary for cross-tissue integration to create a unified cell map covering the entire body. All annotation metadata is on the HCA cell annotation platform, and this is critical for consensus cell type annotation and development of HCA panels for each tissue. We have a full collection of tier 2 metadata for each biological network, and that allows us to translate atlases into new biological and medical insights. And of course, we have a sustainable HCA data ecosystem that allows atlas expansion, drug diagnostics discovery, and training of foundation models. So I want to finally kind of here at the end announce that we are very pleased that we're in the preparation phase of another coordinated publication package. And we have support from Nature Publishing Group for this to coincide with the 10-year anniversary of the Human Cell Atlas and the completion of the HCA version 1 Atlas. And this will highlight the work of the individual biological networks working with the computational community to create and release the first version of atlases. And it'll be an opportunity to publish the source data sets used for integration, to showcase the computational tools and methods used, highlight the impact in the first draft of the atlases, both in advancing biological discovery in terms of insights into cell types, cell states, and also in informing clinical and translational research. And so the biological network coordinators, but also the analysis community, will help guide and curate the publication package. This is an open invitation and call, and in November we'll invite the draft abstracts to be due from the biological networks. In March will be the paper submission deadline, and by July a final draft of perspectives and other content for a target publication date of October. So that's the current sort of plan and timeline. Now, please join me in welcoming Chloe Villani, who will present the recently approved version 1 Immune Atlas. Very exciting. If you have questions for Chloe, please type them in the Zoom Q&A. We'll have time for a couple of questions right after Chloe's talk, and the rest can be addressed at the end of today's session.
Thank you, Sarah. And so it is my great pleasure to speak in the name of a very large team to present to you our version 1 Atlas. This was done across multiple teams, including members of the Immune Bio Network. As Sarah mentioned, this has been done in very close partnership with the Genetic Diversity Bio Network, as well as member of the integration team. So I'm speaking on the behalf of the entire team today, who I'm pleased to introduce you through the lenses of these pictures. We've been meeting weekly for well over 2 years. And so here you have the different trainees that have been involved across multiple groups, including Mata Lucan and Fabian Theiss, Sahat Teichmann's group, Holger Heinz, Shine Prybakar, Gary Reynolds, and myself, and several members on the entire bio network who have been active contributors and giving us feedback. So a quick introduction for those of you who don't know the Immune Bio Network. I have the pleasure to co-coordinate the bio network with Gary Reynolds. We have over 600 members spread across over 40 countries, and we have a large scope, which is to map the human immune system across tissue, development, ethnicity, and lifespan. And immune cells are distributed across all organs. So a couple of years ago, we asked, where should we start? And so we had several engagement activities to ask the community, where should we start? And we came up with 4 tiers of priority through this community engagement effort. The first one being blood, as it's a window in human health. It's easily sample, and it was importantly the type of tissue with the largest collection of available single-cell data sets. And it could help us to think about translational application. The second tier are tied to tissues that are truly tied to the immune system, such as tonsil, bone marrow, cord blood, lymph nodes, spleen, and thymus. The third one being barrier tissue that are enriched for resident memory cells, such as the lung, the GI tract, and the skin. And finally, all other tissues. So we opted, after some debate, to start with blood. As I mentioned, it is a window into human health, easily sampled, many data sets. And we also reflected on the fact that having a really good understanding of circulating immune cells would help us move forward with the next iteration of the atlas, so we could distinguish resident memory cells from circulating cells. We hypothesized there would be a finite number of biologically important immune cell states and program, and some would be conserved across disease conditions. And so for the first version of this atlas, we've gathered over 9 million cells that have passed QCs, over 2,600 samples across 2 conditions. And I'll expand more on these 2 conditions in a minute. The goal is to establish a reference of circulating immune cells. Because we have so many cells and so many donors compared to other biological network, we had the opportunity to optimize our computational framework to build atlases at scale. We also wanted to generate data sets that could be an atlas that could be used to develop consensus cell annotation and start running cell annotation Jamboree, which I'll touch on towards the end of my presentation. And of course, we want to be able to support queries related to the host immune response and how demographic background can shape cellular identities. And so, as Sarah highlighted, these were the different criteria for the version 1 atlas. So in the next few slides, I'm going to show you how we've met these criteria, including incorporating landmark studies, being robust to technical factors, being driven by our biological network, putting some of our annotation on the cell annotation platform, and gathering all the code, data, and metadata. So these are the list of data sets that we have integrated. As you can see, we've mostly focused on healthy and COVID. And there was some debate when we gathered these data sets within the community on whether to do many more disease or to focus on one. And the reason we initially decided to focus on two main conditions is tied to the challenge of doing data integration. When you integrate disease and healthy, ideally you want to have disease data sets that come from multiple independent studies, multiple independent institutions, multiple different countries. And this COVID data set gave us this unique opportunity to test this integration effort across such parameters. And so we have some unpublished and some published data sets. You can see we are across different 10x genomics chemistry with a range of number of cells going from 40,000 cells to over 2 million cells and a range of donors. We ended up remapping all of our data sets to a common reference genome, and I've gathered all the metadata. And in terms of distribution, we do have some of these studies had few other disease involved, which we've also compiled. We're almost 50 in terms of sex. And we have some level of representation across different countries and continents, but you can see some continents are not represented very well. And so, for example, for South America and Africa, the goal is to work in partnerships with the Genetic Diversity Bionetwork to integrate additional data sets from these two continents in the next iteration. And so, for sake of time, I'll just focus on three key aspects of strategies we developed to assemble a version 1 atlas, going over feature selection optimization, hyperparameter optimization, and hierarchical annotation. And so this slide summarizes the strategy to build reference atlas that is robust to technical factors. We spent a fair amount of time thinking about QC strategies. We realigned to a common reference genome. We've done quality control thresholding across lineages. We've optimized our feature selections by looking at both highly variable genes in healthy and COVID and looking at the union of highly variable genes. We've tested over 5 different integration methods and honed in onto 2, Harmony and CVI, on which we did hyperparameter tuning, looking at how to select highly variable genes, PC and latent factors, integration methods, specific parameters, and defining neighborhoods. And so we wanted to use a systematic approach to define which integration method and parameters to select ultimately using both qualitative and quantitative assessments. And so starting with the qualitative assessment on the left, we wanted to look at both technical evaluation and biological evaluation, looking at some of the embeddings. So for example, sources of technical variation included the source of the data set, 10x chemistry, the batches, the cell range version, the reference genome, gender, donor, number of genes, and percentage mitochondrial. So you can see, for example, this embedding, we had batch-specific islands, so that would have gotten us to reject this integration version. We also wanted to assess biological variation because we know so much about immune cells in the blood. So we could look at co-localization of markers for non-cantaceous cell subset and make sure that the clustering was right. For example, here we lack the resolution of IgG from IgA plasma cells, which should be split. So that's an example of us rejecting, for example, a particular integration. In parallel, we also did some quantitative assessment using different metrics, including those incorporated in SCIB, and developed novel metrics based on RNAs to looking at the co-localization of marker genes in the neighborhood graph. So because we know so much of this biology, as I mentioned, we could use qualitative assessment, but you can actually quantify this by looking at cells you expect to be close to each other. So I'll give you an example on my next slides for the CD4 T cells, where we're showing you the Moran's I score for SCVI in yellow and harmony here in blue, where we would expect high level of biology co-clustering. In the context of harmony, you get a lower score, and indeed the cells are colocalizing, and in SCVI, the cells are not colocalizing as well, hence the higher score. And so we observed this across many different lineages and ended up honing on harmony. For the annotation strategy, we wanted to capture the intralineage diversity. And so what we did is we performed fine-grained splits within each lineage to capture biological variability across the millions of cells we had to integrate. And given the scale of our effort, the standard lineage grouping was missing some of the key lineage diversity and was misassigning cells that was leading to inaccurate resolution. And so what we've done is we tried to do some lineage split at high resolution and do then unbiased clustering. So for example, we would split the cells, remove the doublet, switch the misclassified cells between clusters, and do 2 to 3 iterations. And so ultimately, we ended up with 3 million CD8 and NK cells across 33 subsets, 2.2 million cells of CD4 T cells across 25 subsets, 2.5 million myeloid cells across 27 subsets, and 800,000 B cells across 31 subsets, with about 500,000 cells of hematopoietic stem cells and other cell types. And so in terms of the annotation strategy, using that hierarchical approach that I just showed you, we ended up splitting the CD4 T cells across naive central memory effector proliferating the CD8 T cells in similar way, but we also added the gamma delta NK and mate cells. The myeloid cells, we looked into DCs, dendritic cells specifically, and monocyte and DC2, some of which are related, I'll show you. The B and plasma were across naive, memory, and plasma cells, hematopoietic stem cells, and all other cells. And so here I'm showing you an example of these myeloid cells where we did indeed split between dendritic cell, the CDC2, and all DCs. And one of the reasons we kept the DC2 for annotation purposes, they're here, is that some of these gene programs are closely related to activated monocytes. So we want to be captured that transition here. And so as we went through all of these annotation, we also made sure that we capture the technical variability uniformly across all cell subsets. So for example, all of the columns here are the different cell subset defined, and we look at the distribution across disease, the genomics assays, and the different studies, and you can see they were broadly well distributed, and we've done that systematically. And so for sake of time, I'll just give you some quick overview so you can see some of these embedding across 12 subset of naive and central memory, 10 subset of effector, and 11 subset of NK gamma delta. We will be uploading the detailed annotation on the cell annotation platform shortly. We had 10 subset of naive and central memory and 15 of effector that you could see here. And about almost equal split between naive memory and atypical B cells and 37,000 subset cells of plasma cells that you can see here, which will also be uploaded with annotation soon on the cell annotation platform. The code is now will be deposited in our HE data portal, as well as the FASTQ files in the data repository. The integrated object will be available annotated with the HE data repository and eventually on the cell by gene portal. We've also started moving our annotation on the cell annotation platform. I'm going to show you a little video here for the immune cells that will soon be released publicly. So you'll be able to go on the website with the project 648. You're going to have a description of all the different cell types here that are color coded. And you see on the graph here, the distribution. You can explore the data sets. We're going to have the summary of the study and what we've done with some of the metadata here. You'll be able on the left side to download some of the data sets, and you'll be able to explore the molecular data here where it's uploading our final UMAP embedding for the monocytes, as I was showing you. And so you have all the list of the subset here, along with the list of number of cells captured for each, and you're going to have a chance to give us feedback on the annotation. And we'll be hosting some webinars to show people how to give feedback. You'll be able to explore the metadata of why we call a cell a certain way, including the different marker genes. And you'll be able to show on the heat map the expression of particular marker genes we've used to annotate a particular cell type. We're showing you here C1Q8 that was specific for that particular cluster, and you can click on that gene. It's going to show you that particular gene expression here, explaining how we ended up annotating these cells this way. And so standardizing the input of the data and annotation is only the first step. Now comes the real challenge where we need the full community to engage with us, that is deriving consensus annotation. Now everybody will have access to the same 9 million cells, integrated using the same pipeline. And the question is, if we give everyone the same 9 million cells, will we call them the same way? And so this will serve as the basis for the Immune Bionetwork Cell Annotation Jamboree, where people will be able to either do this outside the CAP or using the CAP user interface, which enables us to gather feedback. We'll propose 3 different tasks for the Jamboree. The first one, mandatory to all, will be to define cell subset, including providing the parameters for integration and clustering, and cell labels and marker genes. The second one will be tied to those of you who want to provide solutions for cell spectrum characterization, as we know all too well that immune cells don't have binary identity. They're often part of a spectrum. So inviting people to help define landmarks and propose quantitative readouts of cell spectrum definition, and we'll include additional modalities to help refine annotation, such as repertoire and surface protein. Finally, a third task will be tied to defining coverage for cell subset recovery. For example, we want to assess how many donors and independent studies inside technology processing modalities are actually needed to identify all predicting cell subset within a particular cell lineage. We have the opportunity to do this with the Immune Bionetwork having the largest data sets to do so, and lesson learned here could apply more broadly to other biological network. And so in terms of our timeline, this month we'll be circulating a sign-up sheet. So check your inbox if you're interested in participating. And we'll hope to launch a Jamboree in November, and we'll provide more details. Everybody will have up to 12 weeks to complete the task, and we'll plan for an in-person meeting to compare or contrast our results, which will be compiled in early 2026 to define the areas of disagreement to help us to come together to resolve conflicts, and generate cell cards on CAP that will summarize the areas of consensus. So this is how we met all the criteria. Currently, we're uploading all of our data on the cell annotation platform and prepare and as well as the cell annotation, on the HT data repository and on the cell annotation platform. And once complete, we'll be set to launch our cell annotation Jamboree. So with that, I want to acknowledge the amazing team I was speaking on behalf of, the entire Immune Bionetwork for a contribution. It's been really hard work, and we're really proud of what we've put together. And of course, the Chunk Zuckerberg Initiative, we've provided funding to support this integration effort. And so now I'll stop sharing so we can go back and discuss HCA 2.0. Ellen? Okay. And so we'll talk a little bit about the phase 2 of our strategic goal. So of course, as you heard, we are now hyper-focused on completing every version 1 atlas. But moving forward, we're going to build upon this version 1 atlas and adding 3 additional pillars, which I'm going to describe on the other slide, including spatial, clinically applied atlas, and foundation models, while continuing our mission of building global representation. Next slide. And so continuing on building on version 1, our vision was always to go beyond the individual 18 biological network atlases, but to really create a single unified reference. And so we're going to be working towards integrating all of the version 1 atlas into a single unified reference. We also want to work towards expanding the reference to incorporating spatial data so we can have a more comprehensive view of every organ tissue into the N3D. And we'll be incorporating disease as part of HCA 2.0. Disease represents the normal human in vivo perturbation. They will showcase the clinical utility of the atlas as well as help us improve our global representation, and we'll expand on this next few slides. And finally, we'll be incorporating foundation models, building on all of the high quality HCA data sets being generated, including working on developing AI agents and supporting virtual cell endeavors. Next slide. And so over the last year, we've been working on doing landscaping exercise to understand what are the needs and the challenges our community need to overcome. And we've done this through establishing 4 independent tracks. The first one being the technology track led by Holger Heinz and Jay Shin. The second one being the computational algorithm development track led by Shane Prabhakar and Oli Stiegel. The third one being portal development led by Maz Haniffa and Niels Kellingsberg. And the fourth track tied to atlasing health and disease, led by the biological networks and coordinated by myself. And each track was tasked with defining critical questions, challenges, opportunities that the HCA community could tackle as part of our special program, and where the output of our tracks are forming the basis of our strategic plan. And so what I will do in the next few slides and show you where each of these tracks stand at this point. I'll start with the first one, the technology track. Next slide. And so the goals were to define the technology landscape, shortlist technologies, match the right issue, the biological network, and identify potential partners. Next. And so some of the key activities and features included, thinking about which technologies are capture versus imaging based, which one can work with FFP versus flash frozen, the areas of the spots for profiling, the marker panel size, how quickly we can turn around data, its robustness, performance across all tissues, and their ability to do 2D versus 3D. Next. And so as some of their next steps include, they've narrowed down the scope of technologies that are scalable across many tissues, and we need to perform pilots across different organ system and tissues, working with the biological network. We'll be performing pilots to do this and map out how this can be scaled with the goal of maximizing accessibility and cost efficiency for everyone involved as the data contributors in the HCA. Next. The second track, the computational algorithm development track, has been working on defining algorithm and data analytic challenges to empower the building of these spatial atlases. Next. And you can see on the upper right corner the different members of the task force. So the first step was to think about optimal methods development for gene panel selection, cell segmentation, method for integrating single cell suspension with spatial, and scalable pipelines for massive dataset integration. Next, the second step was to think about downstream type of analysis that would need to be done, including cell segmentation, QC, multimodal integration, and democratized data analysis. And next, with the goal of creating some recommendation through a white paper that will be used to fundraise to enable these efforts and empower our entire community in partnership with some tech companies that can support the data analytics and management for HCA Spatial Atlas. Next, the third track is start to portal development next. And the idea here is to define next the strategies for spatial web portal development by reviewing existing infrastructure to support raw and spatial data, to query data and to visualize it. Next, along with collecting user feedback so we don't reinvent the wheel, and make sure that you are all involved in helping shape up a portal that would be useful for everybody. Next. And so right now they're currently working with different members of the community to map the metadata aspects, which is really key to think about the visualization platform, as well as thinking about file formats and how to index the analysis results, working with the computational track. Finally, the last track next is tied to atlasing in health and disease. And here, the goal was to think about the minimal viable product. And we came up with 2 main tracks. One is to do deep profiling in tissues of a few donors. So for example, think about autopsy cases where you can get large amount of tissue from a few donors and really build 3D skeleton of every organ through strategic tissue sampling, versus a case control approach where we can get very small amount of tissue like a biopsy, but sample and profile hundreds of donors. Next. And we are working with other tracks to try to think about the biological and technical metadata that would be needed for integration. And next, along with additional measurements that we should do systematically, for example, such as H&E staining, protein staining, and perhaps even whole genome sequencing on every samples when possible. Next. And so we've also been regrouping and thinking about how we could leverage our V1 Atlas to develop marker panels, building on consensus annotation coming out of Jamboree. That is why it's so important that you all engage with these Jamborees. And next. And we are working with track number 1, focusing on technologies to explore partnerships with companies for panel design and data generation. And some of the current challenges we've identified by engaging with the community is that most effort have focused on case control study design because of tissue access issues. And we really need to be able to push on larger tissue profile, but there's some technological limitation to it because most technologies don't enable us to do 3D reconstruction. And so the only biological network that I've started doing this so far are Brain and the Development Bio Network. So our goal is to empower all biological network and contributors to be able to do this at scale. We are planning on, you have, next slide, you will have seen 2 different surveys circulating in recent months, the spatial focus one and the disease focus one. We have 200 respondents so far. The first one on the left is about the availability of spatial samples that are ready for spatial transcriptomics analysis. And the second one is disease, which can be done both spatially and by single cell suspension. And our goal here is to have global or regional representation. Next slide. And so here you have an overview on this next slide to show the scope of the spatial survey, where we try to understand the type and source of available human tissue, preservation method that was used, the size of the tissue that were banked, and the size of the cohort. Next slide is going to show you some of the summary of what we've noticed. Most of the samples have been frozen or FFPE. People have done mostly targeted approach, mostly on small biopsies. About 60% of the people have generated spatial data at a cellular resolution. Next slide. In the terms of the disease context, the goal here is to define global and regional priorities for disease research. And so the disease survey enables us to understand the disease you're currently all focusing on, the tissue that were banked, the size of the cohort, the disease that every bionetwork would like to prioritize, as well as identifying disease that may be systematic. So we'll have both diseases that are organ-specific and those that are systemic. And the next slide summarizes some of the key findings of the people that have answered the survey. So a lot of the disease that have been flagged include metabolic disease, cancer, autoimmune disease, and infectious disease tied to high global disease burden. 15% were tied to local or regional disease burden, and 20% were tied to rare disease and neglected disease. We had great amount of response from Asia and Europe, and some in North America, and it's really key. Haniffa, who joined us, and myself cannot stress enough how important it is that you engage in these surveys, because we're using this to identify the samples and the cohorts that will be part of HC 2.0. So you have a unique opportunity to be part of this next exciting chapters by taking a moment and answering these surveys. Next slide, please. And so we'll end before taking questions, Muz and I, with the call to action that was highlighted by Sarah. In the next few months to complete HCA 1.0, when a team reach out to you, please help in contributing data and metadata requests at the biological network. It's really key to be able to eventually build our unified reference atlas. You have a unique opportunity to all help shape our version 1 atlas, even if you were not actively engaged in the integration effort, by taking part to the Cell Annotation Jamboree. You get to help us decide what will be the final V1 products. Please engage. And we really need to stay laser focused on finishing these atlases by October. For HC 2.0, please answer these surveys to let us know what analysis-ready spatial and disease samples you have. We invite you to connect and coordinate with your biological network and other consortium. Our goal here is to synergize with other consortiums that are doing this type of work so we can create complementary version 2 atlases. We talked about defining our minimal viable product, especially a few slides ago. We invite your comment and feedback as we build the strategy. And our goal is really as a community to leverage all the high quality data we're generating to create open source foundation models in partnerships with for-profit companies so we can help advance our missions to cure disease. Next slide. So Muz and I will be delighted to take a few questions before we move to the concluding slides. And Muz, I think we have a few questions. Muz, a question for you, or maybe you and I. Are you from Gamel Akebani? Are you going to develop an interface with tissue cell bioinformatics ontology?
Yeah, thanks very much for that question. I mean, I can start with the answer and Chloe can add further. The plan will be to incorporate some level of ontology, and this is where I think working with other consortia where there are ontologies that have been developed can be streamlined and also used within HCA 2.0 and also HCA 1.0 where appropriate. Chloe, do you have anything more that you would like to add there?
Yeah, absolutely. So for the cell annotation platform, we are already incorporating cell ontology framework, including trying to refine cell ontology when these terms don't exist and try to align with ongoing activities. So that is definitely part of the framework. I guess another question tied to the portals, Ms. from Douglas Kale. With the plans for a 2.0, will the Atlas portal be able to integrate with other portals focused on specific organs? Will other portals be able to access the HE portals to inform the organ-specific community portals? Perhaps I can add something very quickly in that regard and invite you to add to it, which is our HE data portal, which is our central data repository for HE, is their source of ground truth when it's about identifying our official atlases. And through the HE data portals, you'll be able to find hyperlinks to different organ-specific portals, other portals that service our data, the foundation portals. So that's a way to kind of connect the portal, the HE data repository to all these different portals. And I hope we're answering your question, Douglas. But we're also, what we showcase in terms of 2.0, I'll invite Muz to jump in, is also to think about how to better serve the data to our community moving forward. Muz, any additional thought you'd like to add?
Yeah, no, I mean, I think that's a very good question. Clearly, having a portal that's going to benefit the community has been demonstrated time and time again with Ensemble and the equivalent for the human genome reference, as well as the AlphaFold web server. So HCA is completely aware for the need that this is served for the global community to benefit. And this is really what we're working towards. But the world of web portal is also moving fast, very rapidly, in that it is no longer going to be a one portal that serves everyone. And the strategy outlined by Chloe in terms of having modular aspects of the portal that are all interconnected so that the user can actually find the relevant information in an easy and intuitive way is what we're aiming for. And obviously, there are many opportunities to make portals potentially region specific in terms of language, and also the opportunity to have a more chatbot-like communication with the portal rather than the sort of more conventional ways of browsing with a computer as we've been doing.
Maybe two more questions, perhaps. One from Afifa Hamid. Were there any challenges encountered during this entire process? I imagine this is referring to version 1 Atlas that were surprising or difficult to comprehend. I don't know that they were surprising or difficult to comprehend, but of course there were challenges in coming together. I think some of the challenges that we've overcome effectively include defining unified file format so that we can really work towards not only a version 1 atlas, but also towards our goal in HT 2.0 to build foundation models. And that comes along with having standardized metadata framework for what we call our tier 1, which is technical metadata, our annotation that is on CAP, as well as our tier 2 metadata. If you don't have standardized field and standardized file format, it's very hard to build foundation models. And we're very proud to have overcome this with some, detailed documentation that we are happy to share with anybody who asks. These are public. And so that it required our community to come together, both on the computational and biological sides, and to define these fields and to do this in a way that works for all biological networks, since our technical metadata and our annotation metadata are common through all biological network. So we're very grateful for everybody who has participated. Muz, anything else you'd like to add?
Yeah, I just wanted to add, Afifa, that's a very good question that you've asked, but maybe I wouldn't say that was surprising or difficult to comprehend. I think one of the key lessons and reflections for me and Chloe as well, I can speak for her, I think, on this, because we've discussed this many times, is how we build trust in the community and how we develop a culture that will enable us to really deliver such a grand scale project. And the way that HCA works is very different to how research is generally done. And making sure that people really see the value and benefit of what something like the HCA initiative can deliver for everyone, and that this is not just for the benefit of advancing a small group of researchers, but actually really for everyone. And we firmly believe in it, and we aim towards that. And that's why I think having that trust and culture that enables this to happen is really appreciated. And we value everybody's contribution, because this is really for something that's bigger than the sum of its individual parts, bigger than a small collection of individuals. And that's something that we would like to emphasize. And I guess maybe it's not surprising, because it is difficult to get trust, but it's something that we feel passionate about, and we really want to make sure people can trust this initiative.
I couldn't agree more with what Ms. just said, and maybe kind of had a few thoughts. We were so grateful that several contributors trusted the endeavor to share unpublished data sets to enable further the atlas. And it's very hard for, from a practical standpoint, to engage hundreds of people to build the data object. That's not very pragmatic, but the opportunity for everybody to engage is when we launch efforts like Cell Annotation Jamboree. This is an opportunity for everybody to engage in this effort and really invite you to be part in finalizing these version 1 atlas. Everybody has a say, and we look forward to all of your feedback. So definitely please engage. And perhaps, Ms., we may have time for one last question.
Yeah.
Eduardo Rosa Molina asks, are you interested in human tissue ultrastructure in normal and disease states to be part of the atlas? I'm not exactly sure what ultrastructure refers to.
I mean, maybe my interpretation of it is maybe cryo-EM type measurements. And perhaps right now, a lot of what we do will have to be technologies that can scale. And it may be that for a small select tissues or cells, we can actually apply technologies that cannot scale, and then later on integrate it in a, within the kind of like multimodal framework. But by and large, for the at scale data production, we will have to have technologies and platforms that are scalable.
Yeah, I couldn't agree more. And I apologize if we didn't get to all of your questions. Rikan Saragi asked if we had other challenges tied to integrating these atlases with other modalities such as population genomics or understanding mechanisms of action. Ms. is working on answering your question by writing. And if you have other questions that we didn't get to attend to, please send us an email and we'll do our best to answer them. And so with that, we'll move to concluding slides. Ms., would you like to take them?
Yes, I will. Just gonna, yep. So just to, for the last few slides, just to say that we have now got a HCA hub, which will allow members to connect. More details on the HCA hub features are here, where you can search and connect with other HCA members based on location, research interests, academic affiliations. Also post and view job opportunities and participate in the HCA community for interest-based discussion groups, for example, disease. And we really want people to use that. And the next slide is on the HCA Executive Office, the team that are behind the operations and of HCA, and there are all of them, which have been fantastic individuals to work with. Aya, Yoshi, Esther, Bridget, Anne, John, Lucia, Christine, Ellen, and Sam. They've been with HCA and are doing an incredible job, and many thanks to them for building this community. And also our commercial discount program partners, which are 10x, BioLegend, BioScript, Complete Genomics, Element Bioscience, MGI, Pass Bioscience, PixelGen, Nanopore, Scale Bioscience, Singleron, and also VisGen. And once again, please do take part in the surveys to inform the strategy planning for spatial focus in terms of the availability of spatial transcriptomic samples. This will help generate the data that will be valuable to all, and also for disease focus areas with regards to global and regional priorities for disease research. And once again, thank you very much for participating today.
Thank you so much. And please do reach out with any questions you may have. We look forward to engaging with all of you in planning and executing HC 2.0.